Aloha, everyone. My name is Evan Kanoa Carroll. Uh, you might have seen in the program, uh, the title of my project is E Ulu Ka Loko Ia, and that means grow the fish pond. So, quick background, Kawainui Marsh used to be a fish pond, actually, very productive one, and today it is just a protected wetland, full of invasive species, but um, we can restore Kawainui fish pond. And of course, there's obvious reasons to do this. Big one is food, secure food, not imported food, and we can keep the tradition alive. And apparently, it's not the first time this has happened, because in the Mo'olelo of Makale, there was a community effort to restore an overgrown Kawainui. So this is a huge marsh. Restoration is going to be a huge undertaking, but Ka'ulu is already making it happen. So here we are, we're zooming in. Uh, so could we just dump fish in the pond and see how it goes? Um, I wouldn't I wouldn't recommend that. Um, fish need oxygen, food, and space. Neither of the or None of those are a guarantee in a pond, to be honest. So what I did was I kilo or observed first. So right down there is a spot called Ulupō Pond, or that's what I've called it, where some potential fish pond pens are going to get set up. In even more. Okay, so I found that this pond definitely needs some work before you put fish in it. First of all, uh, during the summer, it's about 3 inches deep. Funny story, I originally was going to do like a fish survey in the pond, go snorkeling and see what fish invasive species are potentially in the pond. And I found mud, mud, and mud. Um, uh, but anyways, I also found very low oxygen, which implies a serious risk of suffocation. Um, Upstream is about 7 milligrams of O2 per liter, which is pretty good. But once you get down to the pond, you can see 4 out of the 5 spots that I measured are at a point where it's potentially stressful and may suffocate them. So I definitely need to address that first. And one of the ways you can do that is to increase the flow rate of the pond and the circulation. I don't know how you're going to do that, but got to do it. So, but it doesn't hurt to dream. So I also asked how many fish could we grow if we were able to restore the pond and make it roughly 3 feet deep, which is ideal for a fish pond. So to do this, I'm going to compare primary productivity of the Ulupō pond to a fish pond that is growing fish in it right now, called Heʻeia fish pond. It's down that way. So a quick crash course on why primary productivity is important. They make their own food, usually from sunlight, um, and they're the base and the foundation of the food web. So that's these guys there. Oftentimes they're green. Our kupuna didn't necessarily see them up close, but they knew that a green fish pond was a healthy fish pond. And so it supports everything else. Not everything in the food web is equal in terms of efficiency. The closer you are to the base, the more efficient. So ama, a primary consumer, is a native fish that was often grown by native Hawaiians. And it is for every 100 grams grown at the primary level, you could support 10 grams at the primary consumer level. But if you grew a predator fish like this kaku or barracuda or Papio trevally, then you could only grow 1 gram. So it's 10 times more efficient to grow a herbivore. So I'm going to be focusing just on this pilina today. That's really a foundation of this whole idea of a food web, is the idea of pilina between organisms. So I originally bet on Heʻeia being more productive per acre than Ulupō. My reasoning behind that was being that it's less murky, less turbid, so more sunlight passes through, and it's being actively maintained, whereas Ulupō is still in progress. So one of the things I did a lot this summer was I asked my good friend Google for some advice on stuff. One of those things was how to measure primary productivity. One way you can do that, primary producers, when they grow and produce carbon, they also produce oxygen. And so if you can measure the change in the dissolved oxygen in the water, then you can quantify the primary production. So here's my experimental design for my in situ primary productivity, basically measuring it at the pond, in the pond, to the best of my ability to get an accurate reading. Basically 2 bottles. One has the pond water in it with all the green stuff, and then one has just tap water in it as a control. They're hooked up to 2 devices that measure the dissolved oxygen. So what I found was, I found some evidence of primary production or photosynthesis in situ. Basically, when the sun came out, the oxygen went up. When the clouds were out, the oxygen was going down. However, my control, which had just tap water in it, is all over the place. It's up and down, all crazy. So that leads me to believe that there are other things going on with this experiment that are affecting the dissolved oxygen that is not primary production. So I moved to the lab to control some of these conditions. Took the same bottles, put them under a grow light, and I used stir bars to keep them really well mixed. So here's some example of that data. First of all, 12 hours on, I had the light on for 12 hours. You can see that Ulupō went up a lot, which means lots of primary production. Heʻeia went up not so much, and the control is pretty flat. And when I turn the light off, only respiration is happening. They're just breathing, no more production. So Ulupō went down by a lot. Heʻeia went down less. So basically what I did was so these are all rates of change. So I compiled all the rates of change for all the times that I replicated it. And this is what I found. So this is for when the light is on, the net primary production. It varied a lot, more so for Ulupō in green than for Heʻeia in blue. Overall, the differences between the 2 places were not statistically significant, but the average was higher for Ulupō than it was for Heʻeia. And I think a larger sample size may add some statistical confidence to that. And I also found the same thing for the respiration when the light was off. More negative because there's more of it happening at Ulupō than Heʻeia. So, did some hand wavy calculations based on these numbers that I got to put some constraints on how many fish each pond could support per acre and also individually. So, big reveal, Heʻeia fish pond, 229 pounds per acre per year. This is assuming it's 3 feet deep, roughly, consistently. Obviously, that's not true, but on average. Ulupō, 857. Yeah. So, yeah. So Ulupō is winning by quite a bit, yeah? So I put my bet on the wrong pond. So this was based on these averages. So even though the data was all over the place, Ulupō was almost 4 times higher than Heʻeia on average. So expanding this out to the ponds themselves, Ulupō, if you can get it to about 3 feet deep, just that spot that's cleared can produce 165 pounds per year. It's about a fifth of an acre. If you expand it out, obviously more. And Heʻeia, 88 acres, so you got some catching up to do. 20, about just over 20,000 pounds per year at 88 acres. So I've been told that the managers at Heʻeia don't quite know what their fish numbers actually are. So I'm curious to see how close I am to the actual number. I think maybe one day I'll know. So I'm not done yet. I decided to look closer and uh, put some of my water samples under a microscope. So you can see, this is what that looks like. It's a little bright, but there's a diatom. Oh, I forgot to mention, diatoms are a primary producer. They're an example of a primary producer, and the fish like them. So there's one in the upper left corner, and there's a long, spiny one at the bottom. It's kind of hard to see, but you'll see it later. And you can see some of them move around, but you can tell they're greener than their surroundings, basically. So you can definitely tell that they're producing primarily. So here's some representatives from each pond. I found a lot of Surreyella, some Nitschia, but generally the diversity was higher at Heʻeia. You can see here's some examples from Heʻeia. There's a long spindly one. But the abundance of diatoms was higher at Ulupō. So more diverse at Heʻeia, more abundant at Ulupō, which is good because that means more food for the fish. So the bottom line is Ulupō has huge potential to support native fish once you restore it. And the microscopy also backs up that conclusion. So looking forward, it's important to keep up that kilo. So my cohort and I, we set up water quality monitoring sites in the pond, just so you have a place to measure and keep track of the oxygen. Oxygen is important, but also other factors like temperature. And of course, this opens up a whole bunch of questions like, what about the rest of Kawainui? You know, is it the same? Is it more or less? And what can you do to naturally and sustainably increase the production of the pond? So here we are, back to the pond. Big mahalo to all these people. Any questions?